Okay, great. So, okay, thank you very much for coming to what is now the third talk I've given here. And I promise not to have any quantum field theory in this talk. So, first of all, let's start with a quiz question. So, what actually is it? Anybody got any guesses? What is this?
It's a genome.
It's a genome, yeah. Anybody, any ideas what it is? Well, you know I like quantum field theory, so if you came to my talks last week. So this is the gene for doing Feynman diagrams in 4 minus epsilon dimensions. Okay. So one thing it isn't. I'm the only person who gets that joke. So one thing this isn't. I give this talk to many different types of audiences, and sometimes people say, well, given the title of this talk, they say that so this is the genetic code. And so the reason I show this slide is to check if my slide advancer is working. The reason I show this slide is to say that this is not the genetic code. This is the genome, as you correctly said. Now, so what those ACGTs are, as you all know, are the base pairs. And what I'm going to be talking about in today's talk is a translation. So just to make sure that everybody's on the same page here, let me just very quickly go through that. So here is a DNA. It is transcribed by a machine called RNA polymerase, which makes small transcripts of messenger RNA. The DNA is built from those 4 bases that we saw. mRNA is also built from essentially the same 4 nucleotide bases with one minor difference. And the mRNA produces the proteins by going along by ribosomes going along the mRNA and spitting out the proteins like that. And the proteins are built from the 20 amino acids. So this thing is behaving badly. So what the genetic code is, it's the map that's used by this machine to translate the message from mRNA triplets of 4 bases into the 20 amino acids of life. Okay. So I didn't show the slide, it skipped past it. So this is how the machine works. So this is your mRNA transcript. And the bases are organized in groups of 3, which are called triplet codons. And the machine walks along here. And what happens in translation is a very complicated process going on in the cell where the right amino acid is brought in to the ribosome. The ribosome basically links it up to the nascent protein, and then the protein emerges from the ribosome. So as I said, the genetic code is the map that translates the 4 bases into 20 amino acids. So this is what it looks like, and the way you read it is as follows. You start off with the first position here, the second position here, and the third position by one of these. So for example, if you look at U, U, and U, that codon would code for phenylalanine, and similarly for all the other 20 amino acids shown here. In RNA it's U, in DNA it's T, but basically it's the same thing. It's called the canonical genetic code because it's the same for all living organisms, with very few and very minor exceptions, which we'll talk about in a few minutes. And so it's the same for everything, and that's how we can do genetic engineering. Now the question that I'm going to ask is this. If we rebooted life on Earth, would we have the same genetic code? Okay, so how many people think yes, we would have the same genetic code? Nobody. How many people think, or something very similar to it? How many people think no, it would just be a matter of chance and we'd get something different? 1, 2, 3, 4, okay, the majority. How many people think no? So my answer is yes, or roughly yes. And how many people said no who had seen this talk before? Okay, nobody? Okay, good. All right, so I'm going to argue that it is, that we would see something similar, and I'll explain why. So what I'm going to talk about, the title of the talk, the real title, is Phase Transitions in Early Life, Clues from the Genetic Code. and this is work that was done with a student, Kalinda Sigian, who is now a professor of microbiology at University of Wisconsin, and Carl Rose. Okay, so what I'm going to tell you about in the following hour is this. I'm going to first of all give you some propaganda. So I want to explain to you why studying this is a physics problem. And it's not the usual stuff about, well, you know, physics expands into biology and so on and so forth. The real reason why I want to give you this propaganda is because the question you're going to need to think about is what is the appropriate level of description for modeling. So this was something that was brought up last week in the summer school for PhD students. Alan organized a session on modeling and different modeling philosophies. And I talked then about individual level modeling and statistical mechanics, but I want to now talk about a different type of modeling, a different kind of minimal modeling. Because I think how you model in biology is not obvious, and it's one of the reasons why this is a hard problem to do. So I want to present my view on how one should do that. Now, the second thing I want to talk about is that the genetic code is not a frozen accident. So most of you who voted no, they thought the code would be the same, would be different if you rebooted life. That would be probably most people's viewpoint. And it really first was enunciated by Francis Crick in 1968. And he said the code was a frozen accident for the following reason. He assumed that all life on Earth is descended from a single organism. And he didn't know that at the time, but it turned out to be true. And I'll show you why later. And so whatever genetic code that organism had, we have as well. And the reason that we must have it as well is because if the genetic codes changed, then you would be getting the wrong proteins. The organism would be getting the wrong amino acids, making the wrong proteins, and so the organisms would be suboptimal, or in the worst case, those changes would be fatal. So the code can't evolve, and so therefore it is frozen, and the code is just some random historical accident, and so we call the code a frozen accident. So I want to convince you that that's not true. I'll show you that it's definitely not frozen, and I'll show you that it's not an accident. And the way I'll show you it's not an accident is I'll show you that the genetic code has the following property, that it is essentially optimal in minimizing errors. It minimizes errors in translation, and it minimizes errors that come from mutations. And so I'm going to show you how we know that, and so we're going to have two properties of the genetic code now, that it's universal and it is essentially optimal. Now, once I've convinced you of that, then I have to explain to you how we can get round Francis Crick's, essentially his paradox about it being a frozen accident. So I'm going to show you how a code can evolve in principle, and I'll show you not just the mechanisms, but I'll show you simulations of the evolution, and we'll see that the code can evolve, but only if we make certain assumptions about the nature of early life, and so I'll be telling you what this means for evolution. So you got a question? Yeah.
Yeah, but if you permute just the use of the columns, maybe just for some that are similar frequency in proteins, that would be possible. No, I mean.
Well, we'll see. So then let's leave the whole question to later, because you'll see where I'm going with this. It's more than that. Okay. So I probably won't talk about that. Okay. So most of this talk from here to about here is essentially analysis of data and real calculation. This part here is more speculative, so and interpretive, and that's sort of where we are right now in terms of understanding what this means. So since we're going to end with speculations, I want to begin with a fairy tale. So the fairy tale starts off like this: once upon a time, life started on Earth. And how did it start? Well, nobody knows, but presumably there was some sort of autocatalytic reaction that became the basis for early metabolism. Information molecules learned how to store information, and eventually life became more and more complex until the earliest cells were formed, the so-called prokaryotes, cells without a nucleus, and then life became more complex still until eventually you have the cells with a nucleus, the eukaryotes, which include us. And so that's the that's the story, but it's completely wrong. And the person who really did the most to show that it was wrong is Carl Woese, shown here in characteristic pose in his office at University of Illinois, on the front page of the New York Times, November the 3rd, 1977. Now, why was he there? Well, the headline says, "Scientists discover a form of life that predates higher organisms," but that's not quite true. What he had done, and was published on that day in PNAS, was the mapping out, or at least the first draft of the mapping out of the evolutionary history of life on Earth. And what he had discovered was the three domains of life. In fact, he discovered the archaea. So Carl is no longer with us, but this work is a work that he participated in during his last 10 years or so. Now, how did Carl Woese map out the history of life? So what he did was he realized that if we look at molecular sequences of some part of the cell that is in every cell, then we can look at the changes in those sequences over evolutionary time and map out the history that way. And so his chosen instrument of biological revelation was the ribosome, the machine responsible for translation, because every cell has to have that. So he set out to sequence parts of the ribosome. What we do today is we look at the 16S ribosomal RNA gene, and here's a bit of it shown here. So this is human, yeast, corn, E. coli, various other organisms. Down here is Sulfolobus, an archaeon. And these sequences are very similar, but there are slight differences. And so from those differences, those differences reflect the divergent evolutionary history of these organisms. And what people try to do is construct some sort of edit path between these sequences, and that edit path is then interpreted as the evolutionary history of organisms. And there's different criteria that people use to do that: maximum likelihood, parsimony, nearest neighbor, and so on and so forth. So that's an art in and of itself, but the outcome of that is shown in this picture here from Norman Pace's review in Science, 97. So there are three domains of life. This is what got Carl on the headlines. So the bacteria, the archaea, and the eukaryotes. So there's you. Virtually everything else on this tree, which is now much bushier than it was then, is microbial. These things here are the things that were called prokaryotes. So if you look at them under a microscope, they look like round blobby things. But if you look at them genetically, you see that these things are as different from these as you are from these. And so we understood that the history of life was really not what we had thought. Now, this point here is the root of the tree. It's nearer the bacterial branch than the archaea eukaryote split, and that organism has got a name. It's called LUCA, the last universal common ancestor. So basically, sorry about this. What is going on today? Right. Okay, so this is what you see in some of the older textbooks. This is what we know today, and the reason it's wrong is that these prokaryotes are not in any sense predecessors of the eukaryotes. They evolved along with them on the parallel tracks, like you see there. Okay, now one of the things that Woese and Fox also commented upon in their early paper was the time issue. So this is their paper in PNAS, and what they noticed was if you look at the organisms near the root of the phylogenetic tree, such as they could determine it in those days, they identified those as being basically cyanobacteria and essentially of order 3 billion years old or somewhat older. So therefore, you have an interesting paradox, because you know that the world is 4.6 billion years old. So if the oldest organisms, or the last universal common ancestor, is say 3 or 3.5 billion years, today most people would say it's 3.8 billion years, then that means that life had to go from nothing, absolutely nothing, to essentially the complexity of the modern cell in less than 1 billion years. And we know it had to be the complexity of the modern cell because otherwise it had to have ribosomes. And we wouldn't have been able to track the existence of that phylogenetically if it didn't. So life didn't start at the root of this tree. That's just the end point of the beginning phase of life. So the question is, how on earth could it go from nothing to essentially the complexity of the modern cell in less than 1 billion years, maybe a lot less, and then in the remaining 3.8 billion years, nothing very much happened. Well, okay, you made a few dinosaurs and humans and things like this, but essentially the cell itself didn't really evolve much in essential terms. So that was the paradox. So the other thing that this tree does is it shows you that all life on Earth is related. We didn't know that before. And the question that I want to talk about is why can't we see further back in time? Now, you might say, well, this whole molecular phylogeny business is a miracle that you can do it at all, and maybe the signal just peters out, and so that's why we can't see further back in time. But I'm going to argue that there's a fundamental reason that we can't see further back in time. And that fundamental reason is to do with an ingrained assumption that you all have, and that is that we can represent families by family trees. So you are used to making your own family tree. You look at your children, your children's children, and so on, and you transmit your genes to them, and you can build a family tree in the usual way. But that is not mandatory. That assumes that we transmit genes, we'll say vertically, to successive generations. Well, how about if we transmit genes horizontally? Okay, if you have horizontal transfer of genes being transferred to non-related organisms, then you won't get a tree at all. You'll get some kind of network. And we're going to see that that's going to be an important part of this puzzle. Now, when we talk about evolution, Darwinian vertical evolution, what we usually mean by that, the way we understand it, is in terms of population genetics. And that's very good at answering certain questions, perhaps the origin of species, maybe, but certainly the change in frequency of gene alleles in the population. But what we're interested in here is the nature of evolution before the last universal common ancestor. When you didn't have species, certainly at some point you didn't even have genes. And so we really want to understand a bit more, what drives the evolution of complexity? Why was the process of evolution in early life so fast? And also, is that process manifested and connected with evolution today? So I think there should be a broader view of evolution, of which population genetics is a subset. But there's certainly much more to evolution than that. And I think that's not a very controversial statement. People study chemical evolution, for example, and so on. Okay, so if there's a lot to be said about that evolution as a problem in non-equilibrium statistical mechanics. And so I've written a review article with Carl in Annual Reviews of Condensed Matter Physics, which came out a few years ago. And that describes the sort of complexity and evolutionary dynamics in a more general sense in a lot more detail than I want to go into today. Now, the next thing I want to do now is talk about why this is a physics problem and why that can help us model the genetic code. So I want to talk about the right level of description to understand the evolution of life. Now, as you know, I'm at the University of Illinois. How many people in the room know who this person is? Okay, his name is John Bardeen. Anybody here ever heard of him? He did. He won twice as many Nobel Prizes in physics as Albert Einstein did. Okay? His first Nobel Prize was for inventing this thing, transistor. Yep. And his second was was superconductivity. So let me tell you about superconductivity, because I used to work on superconductivity before I did biological physics. So this is what a superconductor looks like. This is a particular one. It's a great puzzle. It's the high temperature superconductor YbCO, yttrium barium copper oxide. It's got a very complicated crystal structure. And when you look at its resistance as a function of temperature, what happens is that the resistance goes down as you lower the temperature until you get to about 93 degrees Kelvin, and then it drops to 0. And it's literally 0. Okay? I mean, it's not like a very small number. It's as close to 0 as we are able to measure, and that phenomenon is called superconductivity. Now, there's a lot to say about superconductivity, and I don't want to give you a graduate course in it, but the message I want to talk about is that to understand superconductivity, it's not about the atoms. So you might think that how would we try to understand this puzzle of superconductivity? The phenomenon was discovered in about 1911. It wasn't solved until 1957. And some of the phenomena of superconductivity weren't even discovered experimentally until the middle 1960s. Okay? So it took a very long time for this problem to be solved and for all the ramifications to be understood. Now, why? Well, the reason is that at the most basic level, you'd say, well, I've got to look at the atoms. I've got to look at how the atoms are formed in a crystal. I've got to look at the quantum mechanical orbitals and the overlaps between them, try to understand electron transport in the material. And it becomes a very complicated problem, and then you have to see how can I get this flow of electricity without resistance transfers. It turned out that that's not how you solve the problem. So how do you solve the problem? Well, the solution is known as the Bardeen-Cooper-Schrieffer theory of superconductivity. And here's an example of how good it is. So the theory says that superconductors form some new kind of ground state, quantum mechanical ground state, which I'm not going to describe. And that ground state is characterized by having an energy gap. If you want to excite it, the next energy level, it has a gap above that ground state, and that gap at zero temperature is called capital delta. And what the theory predicts is if I take that capital delta, divide it by Boltzmann's constant, times the critical temperature, 93 in the case of yttrium, barium, copper oxide, that will be the ratio, that will be the pure number 3.5. And if you plot that energy gap as a function of 3/2 kTc for materials for which the theory should apply, they fall nicely on the line. These materials here aren't describable by this simple version of the theory. So that's very interesting. If you look at this energy gap as a function of temperature, what you find is if you plot T over Tc, you find that the data fall onto one universal curve, and that curve is identical to the curve predicted by the theory. So that's just one of many, many predictions that the theory makes. And the predictions are the same for all materials to which the theory applies. Now, the big surprise is that this model leaves out much of the physics and all of the chemistry. The way it was solved was not by worrying about the atoms or phonons or the electronic band structure. What BCS realized was that the only thing that's important is how pairs of electrons dance around each other on the crystal lattice. And that correlated motion of the electrons, when you just describe that, turns out to predict all these results. So what they look at is the cooperativity of the hopping electrons. That's the important thing. So you get something for nothing, it seems, but not really. Because if you say to somebody, well, why is the critical temperature of yttrium, barium, copper oxide 93 Kelvin? The answer is, don't know, we can't predict that. But if you ask about properties like the energy gap and so on and other things, we can predict those very well. So some questions are good ones to ask because they don't depend on details. Other questions, like calculating Tc, are bad ones to ask because they do depend on details. So the moral of the story is focus on the process and not the realization. Now, what I mean about that? Well, process means the quantum dynamics of correlated electrons. That's the important thing. The realization is what you need to do in the laboratory to make something that does this. So in the case of the high temperature superconductors, you mix up yttrium, barium, copper, and oxygen, stir them up in the lab, shake and bake, and you can make a high temperature superconductor. Even a high school kid could do it. So you can't explain superconductivity by just thinking about atoms. You have to understand their cooperative behavior and focus on what really matters, which is the cooperativity. So for superconductors, it's not about the atoms. And I claim that for life, it's not about the molecules. Okay, now, when I go to a workshop on, say, evolution or origins of life or something, somebody will stand up and they'll say, life is chemistry. Okay, and everybody will nod, because it's a very non-controversial statement, and then they'll start talking about lots of biochemical pathways and so on. And I completely disagree. I don't think life has anything to do with the molecules. Because I think that life is about the evolutionary process, and the molecules are just the realization that you need to mix up in a lab to create something that manifests the process. I don't think you can understand life by just thinking about the atoms that make us up, because they also make up inanimate objects. It's the interactions and the correlations and the cooperation between them, between the atoms and the molecules, that creates living organisms. So why is that important? When we try to model something like the genetic code, I'm going to try to make a minimal model a model that's kind of like the BCS model of superconductivity, one that just focuses on the important details and doesn't even try to describe things like chemistry and things like that, which require much more detailed understanding. Okay, so what really matters in biology are collective effects, and I want to now talk about microbial ecosystems. So this is going to be important, not just because people here are interested in microbial ecosystems, but because it's going to be important for understanding the origin of life. So here's a phenomenon that probably many of you know about, and that is horizontal gene transfer. So let me just try to explain the rationale for this. So let's try this in this audience, let's see if this will work. How many people here have seen the science fiction movie, The Matrix? Okay, everybody has, well, except Aaron. Well, okay, well, you'll get your chance a little bit later. So in that movie, there's a very exciting moment where one of the characters has to escape from a very dangerous situation. And the only way out is to fly a helicopter. But the problem is she's got a helicopter, but she doesn't know how to fly it. So what she does is she calls up to her sort of mission control and says, beam into my brain the instructions for how to fly a helicopter. So they do that, and then she just gets in and flies away. Okay? Now that's science fiction, and that doesn't happen to us. If it did happen, and you had a burning desire to learn about quantum field theory or phase transitions in physics or something like that, I could just sort of pop out that gene I showed you at the beginning, the 4-epsilon integral gene, and you could just absorb that gene, and then bang ho, you'd suddenly know how to do the normalization group calculation. Now that doesn't work in the real world for us, but it does work for microbes. Okay, they do that. They swap DNA, and they can express the DNA that they receive, and so they can learn new tricks. Now the way they do this is through 3 mechanisms: transduction, conjugation, and transfection, or transformation. So this is really just sucking up free DNA from the environment. This is bacteria having sex, so they can get close to one another. They can transfer little plasmid loops of DNA from one to the other, and they can be incorporated into the new cell and can be expressed. And the other way is viral infection. So here what might happen is a virus will come in, it will inject its DNA into the cell. That DNA will, in some viruses, will go into the genome, the chromosome of the cell, and maybe replicate along with the cell for maybe many generations. But then sometime later, if it is stressed, that DNA, that prophage as it's called, will come out. And then what happens is that DNA will start building lots of baby viruses, and those baby viruses, maybe 100 or so of them, will be grown inside the cell, hijacking the cell's own replication machinery, and then they will pop out, they will take the DNA with them, their own DNA, and go and infect another cell. But when they take their own DNA, they also take neighboring segments of DNA, and so genes can be transferred between cells by this kind of chain reaction process. Now, microbiologists have known about these for many years, but the question that someone like me is interested in is a sort of statistical question or community level question, is that if the microbes can do this pairwise, what happens when whole communities do it? Okay, so we really didn't know the answer to this until a few years ago, when it became painfully evident that this was a real disaster, and the disaster I'm talking about is the spread of antibiotic resistance genes. So we've known about horizontal gene transfer since the late 1950s. Earlier we had started using antibiotics, and what came as a big surprise was that within a few years of introducing a new antibiotic, essentially it starts to lose its efficacy because of horizontal gene transfer of antibiotic resistance genes. So the evolutionary timescale is 5 years. It's not millions of years, and it's accelerated because of horizontal gene transfer. Okay, here's another example that I think is a beautiful one. This is work of Forrest Wall and Penny Chisholm's groups, and this is to do with this thing here, which is a cyanophage. So this is a phage that infects photosynthetic cyanobacteria. And the interesting thing about this phage is that it carries photosystem 2 genes with it. So, so the question you might want to ask is, well, why would a virus, why would a phage need photosynthesis genes? Well, the answer is very interesting. Think about it. The virus can infect a cell, and it'll produce, while it's killing the cell, it'll produce more baby viruses. But suppose it could keep the cell alive for a little bit longer. Then the cell will produce more viruses. So what it does is it essentially carries along with it its own life support system for the cells. It carries the photosynthesis genes, so it can sort of set up a second version of photosynthesis inside the cell as it is producing viral copies, and so it makes more baby viruses. So that clearly benefits the virus. Now what this phylogenetic tree here shows, this is for one of these genes, is that these genes have been swapped back and forward between the virus and the host many times over evolutionary time. And the reason that has happened, well, it's happened because of horizontal gene transfer, but the effect of that is to benefit the bacteria. And why is it benefits the bacteria? Well, the genes can diversify much faster in the phage than they can in the host, because there's no error checking and so on. So what that means is that this gene can essentially mutate, and possibly improved versions of it may be created in the virusphere, and so the phages provide a huge genetic reservoir for their hosts, and that gives an advantage to the bacteria, to the cyanobacteria, in terms of invading new ecological niches. In the case of cyanobacteria, they can tune the wavelength that they receive, and so they can go to greater depths in the ocean, and so it's a competitive advantage. So basically, the message is that the cloud was invented 3 or 4 billion years ago by bacteria, and it is an important part of their evolution. Okay, so that's how this community can be essentially stabilized and made more resilient by these macrophage interactions. Okay, so now having said that, I want to talk about the genetic code again. So I want to first of all tell you about patterns in the genetic code. So here's the canonical genetic code, one you've seen before. Here is the yeast genetic code, but this is the genetic code for the mitochondria, not for the nuclear code. If I just toggle back and forth, you can see it's basically the same. Basically the same, but with very slight differences. So this code has managed to evolve in the mitochondria. Now, not very important in mitochondria, because the mitochondria get very few of the genes are actually important, because they mostly are parasitic on the nucleus. But it does show you that the code is capable of evolution. Sorry about this. What's going on today? Okay, the next thing I want to tell you about the code is that it's degenerate. So methionine and tryptophan, shown here and here, are the only amino acids with one codon. Everything else is degenerate. And they cluster, so they're not scattered randomly, but they occur in blocks. So this way of representing the genetic code is biologically significant. And if you look at the chemical properties, what you find is most hydrophobic amino acids are over on this side of the code, and most of the most hydrophilic ones are over on this side of the code. So there's a gradation of chemical properties of the amino acids throughout the code table. Now this was first noticed by Carl Woese in 1965, 1966, and he devised a way to quantify this evolution of the chemical properties, and he measured it, and this measure has a name. It's a very bad name, and it's called the polar requirement. Okay, the polar requirement is a sort of counterplay between polar and non-polar interactions of the amino acids. But for our intents and purposes, it just measures the balance between hydrophobicity and hydrophilicity, and essentially it's a number, that's all you need to know. It's a number that is small over here and large over here. Okay, so that's the polar requirement. Now, what I want to tell you is that the genetic code isn't just universal, it's nearly optimal in minimizing errors. And it's optimal in minimizing errors if you use a measure that is based upon the polar requirement. Okay, so let me tell you about that. So the idea was this. if you have point mutations, they can screw up the amino acids that you would get, and therefore screw up the proteins. If you make errors in translation, they will also mess up the sequence. So if you were asked from scratch to try to devise a good genetic code that is robust towards all the noisy effects of biology, what you would try to do is say, okay, let's make it so that if I get the wrong amino acid, at least the amino acid I get is roughly similar to the one I should have got. And maybe then the organisms will be able to survive. Now, you might say, how do we look at that? How can we test that? We've only got one universal genetic code, so we can't look at a bunch of genetic codes and compare them, but we can, and we can do that by doing a Monte Carlo simulation. So here's the idea. Let's take the genetic code table, and now let's permute where the amino acids sit. So how many ways are there to do that? Well, there's 20 factorial, that's about 10 to the 18. So obviously we can't enumerate all of these, but we can sample them by Monte Carlo simulation. So we can generate a whole library of synthetic genetic code tables. Now what we want to do is we want to construct a number, a measure, a figure of merit that tells us how good each of these synthetic genetic codes is at minimizing errors. Some will be better than others at doing that, and we want to quantify that. So the way to do that was worked out by Hagen Hurst originally in the middle 1990s. And so the basic idea was essentially to sum the differences of the squares of polar requirement numbers for individual places in the code table. So basically what you do is you take a code, you swap the amino acids, and then you look at the difference of the square of the polar requirement numbers, and then you add those up for all positions in the genetic code table. And when you've done that, at the end of it, what you've got is a single number. And if that number is small, it says that the genetic code is very insensitive to making swaps of the amino acids of neighbors. If it's very large, then it means it's very sensitive to that. And it is, the way that you construct this measure is more complicated than I've explained to you, but that will do for now. Now, how do you process that information? So now what you do is you do a Monte Carlo simulation. So you now go and measure that number over all as many codes as you can sample. And what you're going to get then is a code property, in this case the polar requirement score. And this is the probability distribution of that. And what you're going to get is some kind of bell-shaped curve that looks like this. Now, if the genetic code is a frozen accident, then what you'd expect is that the actual value of the genetic code would be like I've shown you here, just something that's in the middle of this distribution. If, on the other hand, the codes you get at random look like this, but the actual genetic code is way out here in the wings, at a much smaller value, then you would say that it's not what you would get at random, something else must have happened. So this was done, this is a paper by Freeland and Hurst from 1998. They were able to simulate 1 million codes, and the title of their paper is The Genetic Code is 1 in a Million. When they simulated 1 million codes, here's the actual value of the score for the canonical genetic code. Here's the distribution that they got, clearly undersampled, but in 1 million codes, they couldn't find one that was better than the canonical genetic code. And of course, this has been improved and the analysis extended and so on and so forth. So I don't want to go into the details of that. So what I've told you up to now is that the genetic code is universal, and it is very, very good, if not optimal, close to optimal, in minimizing errors. So now in the last third of the talk, I want to tell you about how it got that way. So I want to tell you about the mechanism for the evolution of the genetic code. Now, there's a real detailed calculation here, but if I just showed you the calculation without explaining it to you, it would mean nothing. So what I want to do now for the next few minutes is walk you through some sort of examples to give you a kind of physical intuition about how the code has evolved and why it has got the way it is. And then I'll show you the results from detailed calculation. So the puzzle then is that the genetic code is a complex trait. It's a table that is an abstraction of all the processes that go on inside the cell during translation. It has evolved, and yet it's universal. Why isn't there a diversity of codes? Now normally if you have some complex traits like IQ or height or something like that, you get a continuum of values. But that's not the case here. We've just got one code. So why is that? So is the universality accidental, or is it there for a deep reason? And I'm going to claim that it's there for a deep reason. And the reason is that the code is an innovation sharing protocol. It's a protocol, kind of like an operating system of a computer that performs a very special function. And I want to explain about how that works. Now, to talk about how codes evolve, I want to do a thought experiment and appeal to the people who are the real experts in codes. Now, who are the people who are really experts in codes? They've been in the news a lot in the last year or so. They're not biologists and they're not physicists.
Hackers?
Well, hackers maybe, yeah. That would be one description of these people, yes.
Spies.
Is what?
Spies.
Spies, yeah. Spies, like the NSA or CIA or something like this. So I'm going to do a thought experiment. So these people, does anybody recognize these people?
Yeah.
Okay, well Alan does, because he didn't know the answer to the question about the matrix. So basically anybody who knows the answer to the question about the matrix may not know who these people are, and vice versa. So these people are spies from bad 1960s or 1970s TV shows. And so I want to do a thought experiment where one of these spies is trying to send a message to be interpreted through a code book to another spy, another member of their distinguished fraternity. And we're going to try to do a thought experiment to see how codes are actually transmitted in practice. Okay, so let's start off then with this very simple situation. So Emma Peel has a message. She wants to transmit that message to Mr. Powers. So she transmits it, he gets his code book, and then he translates the message. And so far, everything works just fine. So this is like a real message, and the important thing about it, that there's only a single message. So here's the message, translates it, here's what it means. Everything is one-to-one. But Austin Powers is a very careless individual, and so he loses half his code book, and now the message doesn't make any sense to him, although it might make sense to some of you. And so what we learn from this is that this code is a very fragile. If you make a change to the code, or it evolves in some way, then the message instantly becomes garbled. That's basically the insight that Francis Crick had with this frozen accident. But suppose instead that we had a statistical code book, one that doesn't produce one output, but produces a whole spectrum, a whole distribution of outputs, and then the one that is the right one is determined by the actual context. So in this particular example here, Austin Powers has used his statistical code book, and he's come to the conclusion that all the messages are nonsense, but except for this one, and he figures out with a probability of 0.9 or something, I just made it up, that's the message. And so the message is not lost when you have a stochastic code book. So what's important here then is you have a probabilistic code book, and what that means is that you, in the context of translation, is that you have statistical proteins. So let me explain to you what that means again by another analogy. So let's suppose that you think about the translational process today. It's very precise. It works extremely well. It produces the right proteins for you. But what about billions of years ago? What do you think early life was like? Well, think about automobiles as an analogy. So today's automobiles are very carefully constructed. Like in America, the most popular automobile is a Toyota Camry. Okay, it's a very nice machine. It's built with Six Sigma precision, whatever that means. Very, very precise and works very well. On the other hand, if you look at the earliest cars, say ones made by Benz and Mercedes and people like that in the turn of the 19th century, those automobiles, their top speed was 10 miles an hour. Bits of them would fall off all the time. They really couldn't go very fast. They were very bumpy rides, and they really weren't very good. But let's suppose you drove around to your friend's house and the wheel fell off. Well, it's no problem. You just get a bicycle wheel that he has in his shed, get your toolbox, get some nuts and bolts from the basement, screw the wheel back on again, and off you go. That was fine, but if you tried to do that with a modern Toyota Camry, well, good luck. You wouldn't be able to repair it in your own garage. I mean, in America, we can't even build them in Detroit, so there's no way you're going to be able to repair one yourself. So think about early life as being like the early automobiles, something that was like a jalopy, very, very floppy, very imprecise, didn't work very well. If it got the wrong proteins, didn't really matter. As long as the proteins had more or less the right shape to fit together in some way, that was probably good enough. The life wasn't very effective, but as life gradually evolved in complexity, the requirements that were made on the proteins became more and more severe, and so it became more and more important that the translational process became more accurate. And so what that suggests then is if you had a translational machinery, a ribosome, that didn't just produce one protein, but produced a whole suite of proteins, a whole suite of amino acids, just worked kind of at random, but more or less gave you something right. Then as the system became more and more complex, what would happen was because you've got a statistical translational machinery, you're still able to evolve the code and be able to select from the proteins that are produced the right ones for the task. So what that says is that the code can evolve, not by itself, but it can co-evolve along with the complexity of the organisms. And that's the basic insight for how the code it does not get trapped into the frozen accident hypothesis. Now, how does selection pressure influence the code dynamics? So let's think about a different thought experiment. Let's suppose that the National Science Foundation has a call for proposals, and they say, okay, we want a proposal, but it has to be a collaboration between a physicist and biologist, or a mathematician and biologist. In America, at least, people want to encourage more quantification of biology, and so those things come out all the time. So the problem is this, that you sit down to write a proposal with your best friend, but your biologist friend likes to use Microsoft Word, and if you're a mathematician or a physicist, you like to use TeX or LaTeX. And so what might happen is I might sit down with my friend, and she sends me her material for the proposal in Word. I send her my material for the proposal in TeX, and she doesn't know what to do with my TeX. I don't know what to do with her Word documents. And so we squabble, we never manage to figure out how to work together, and we never submit our proposal. Meanwhile, somebody else is writing a proposal, and may not even be as good a proposal, but they start off right away working in Microsoft Word, they submit a proposal, and it gets selected. And so what happens is next time I have to do this, well, I just forget about TeX, we just write a proposal in Word. And this is the sort of thing that has actually happened to me. So this shows you how selection pressure, even though it's not exactly operating on the phenotype, is only operating on the phenotype, can still influence the dynamics of the code. This is the sort of Baldwin effect. Okay, now the next thing is I want to tell you about is competition between innovation sharing protocols. So I'm sure you recognize these people. So this is Bill Gates with his Windows computer here, an early version. This is the late Steve Jobs with a Macintosh computer. And as you probably know, I know something like, I don't know what the numbers are exactly, but something like 95% of the world's computers run Windows, and only a few run the Macintosh operating system. Now, the reason for that is very simple, that the Windows computer is simply better. And in fact, I know Alan and many other people here use a Mac. I use a Windows computer, and the reason that it's better is that it crashes all the time. Well, that's embarrassing. That's a blue screen of death. I see there's a lot of Mac people here, so they don't know what that thing is. You've never seen that before, yes. So even though Windows computers crash all the time, why are they better? They're better because any program that you want to run is available for you on the Windows platform. But if you want the program and you're using a Mac, well, if it's a very well-used program, like say Office or something like that, you might get it. But some of the lesser-known programs you won't find. And so what happens is that basically anything you want, you can get on Microsoft, and the coders are given for free the coding instructions and the developers kits. But for Macintosh, it's very different. You have to buy it, and there's less users, so there's no incentive for a developer to write programs for a Macintosh computer. This became a really big issue when the iPhone came out. So before the iPhone came out, Steve Jobs understood this problem, and so he switched the chip that goes inside Apple computers to be the same chip as the Windows computers use, so that you can run Windows programs on Apple computers. And then when he introduced things like the iPhone and the iPad, what was the big selling point of the iPhone and the iPad, apart from the fact that they actually worked? What did they use in the adverts? What's the big thing about the iPhone and the iPad? Nothing? You don't have them. Anybody here heard of the App Store? Okay, right? There's a billion apps for doing anything you want, and on the competing system, which is Android, there's only or at least at one time there was very few apps. Now they're more or less the same. But for many years, the competitive advantage of the iPhone and the iPad was the App Store. So what you've learned from this example is that the community with access to the broadest range of innovations has an evolutionary advantage. Anything that you want, any gene that you want, more beneficial traits are available to you if you are part of a large code base. And so organisms which have the more popular codes are going to be more protected against invasions from organisms with different codes, and are more likely to invade other niches. And so basically the biggest code base is going to win. Okay, so what we did was a calculation. We did a calculation of, neglect that, of digital life. So what we did was we simulated organisms in a computer, and these organisms are like computer viruses, but they are a bit more sophisticated. So these organisms have a genetic code, and that genetic code is used to translate an underlying genome. They produce from that proteins as instructed by the genome. The proteins contribute to the fitness of the organism, the reproductive fitness, and the organisms then compete in some fitness landscape. Nothing I'm going to tell you depends precisely on details of this. This is a sort of a generic description of what happens. I'm going to skip, in the interest of time, the detailed description of the model. Let me go to the verbal description. So the population is asexual, evolving, reproducing digital organisms. The fitness is a function of the phenotype, and the phenotype depends on the distribution of proteins of the individual. Proteins are obtained by translating a genome with code, with error, and the organisms have a reproduction rate, and they can also create errors. So what do we do then? We run the simulations and we see what happens. Now what do we measure? What's the output of this big digital life calculation? Well, what we're trying to understand is optimality and universality. So what we measure is the error minimization aspect of the code. So what we look at then is we look at the polar requirement scores of between neighboring codons. We look at the average amino acid distance between neighboring codons. If the code is minimizing errors, that distance should be small. If that distance is large, it means that the code will not be very good at minimizing errors. And then universality is basically how many codes are present in the population and how different are they. So we measure the average distance, the Hamming distance between codes in the simulation, and we just simply count how many codes we have. Okay, so here's the results. So on the vertical axis here, I'm plotting the quality of the code, in this case the average amino acid distance between neighboring codons, and along the horizontal axis is time. And then we're going to do the calculation in two different ways, one with regular vertical Darwinian evolution and one with horizontal gene transfer. This here is a calculation, a Monte Carlo calculation of how likely it is that the genetic code that we get could have arisen at random. So just as I showed you the Hagen-Hurst calculation for the optimality of the real genetic code, the canonical genetic code, so we'll do that same Monte Carlo calculation for the codes in our computer simulations. So here's what we find. When we start this simulation without horizontal gene transfer, we see that the codes eventually evolve to become better by the mechanism that I explained. But then what happens is it stops, and then nothing much changes after that. And then you run the simulation again, and so now you get a different frozen accident. You say you get this one over here. And then you run it again, and maybe you get this one. So, and these final code values are not very different from what you would get at random. So they're not unique, and they're not optimal. On the other hand, if you run it with horizontal gene transfer present in the dynamics, then what happens is the codes keep improving, they never stop like here, and eventually they settle down to some asymptotic value, which is very different from what you would get at random.
What determines these asymptotic values?
What is the what?
What determines these asymptotic values? Are they just a function of your starting?
It's a function of
Yes,
of whatever parameters you have in a simulation, but nothing I'm telling you depends on those. So I mean, the precise numbers here and here will differ, but the message is the same. So what you learn from this is that by including horizontal gene transfer of translational machinery in the simulation, you can obtain optimality, but otherwise the system doesn't do that. It gets sort of stuck in local minima. And so this is the sense in which the outcome is reproducible. It converges to a dynamical fixed point. If you now look at the evolution of code distances, so this is the average code distance as a function of time, again, the story is the same. So if you have only vertical gene transfer, you very quickly get locked into some kind of frozen accident. But with horizontal gene transfer, the codes get more and more similar until eventually you have one code left in the simulation. So what happens is then horizontal gene transfer leads to universality. So now I want to talk at the end, just for the last few minutes.
But why is universality necessarily optimal?
It needn't be, but we see that it is. We see that you see that the code evolves to both universal and optimal. It's not obvious. It's not mandatory. Hey, can I? So limits of time. Could I just finish the last couple of minutes? Okay. So I want to now talk about the speculative part of the talk, or more speculative, if you like. So I want to just explain what we think this means for early life. And so this isn't something where I can show you a detailed calculation. So the claim is that polar requirement sees further back in time than sequence phylogeny, that we're understanding something about life before the last universal common ancestor. And the reason we know this is the following. The question is polar requirement special? What happens if you redo Hagen-Hurst's Monte Carlo calculation with something other than polar requirement? So Rob Knight in his PhD thesis with Laura Landweber in 2001 did that exercise, and it's in the last few pages of the appendix of his PhD thesis. He worked on many different properties that he listed here, and for which biochemists can quantify in some way. Properties of amino acid binding with early peptides or proteins or side chains, various things like this. He considered all of those, reran the experiment, and what he found was that nothing worked. That the polar requirement and something very similar to it, which is called Grantham polarity, those free amino acid properties are the ones for which the genetic code is something special. For anything else, it doesn't seem to be very special. Now, what's interesting about this is that these are properties of free amino acids, and so we interpret that as meaning that this is a relic of very early life, before amino acids were bound into macromolecules. In other words, think about it this way. Everything in biology came from something else. What did the genetic code do before it was the genetic code, before you had translation? Well, it was some machinery that was co-opted for translation, presumably some sort of nucleotide catalysis or something like this. So this idea then is that this free amino acid property was something that was baked in very early on in the development of the genetic code, and so is a relic of life before the last universal common ancestor. Okay, so what have I told you in this talk? So I've told you the phase diagram of life. Here is the bacteria, the archaea, and the eukaryotes. Here's the last universal common ancestor. This is our cloud of ignorance surrounding what happened back then. So what I've told you is this. If early life created a set of organisms, all of which had various different code bases, various forms of genetic code, those organisms are able to tolerate ambiguity. If you put a new part in, they'll still work, but there's no global horizontal gene transfer. These organisms compete, and their code bases compete, and eventually one code will win, and you'll have the first universal common ancestor. From that point on, you now have a universal genetic code where there is global horizontal gene transfer, and you have organisms that are capable of tolerating new innovation. So what you're going to get is a communal state with explosive growth and innovation sharing, just like the internet was in 1992 with the introduction of the hypertext transfer protocol on top of TCP/IP. So then all the computers in the world could suddenly talk to each other, and boom, you have the exponential growth of the internet. Now what happened was at some later time, sometime around 3.8 billion years ago, this got cut off, and we transitioned to an era where essentially the translational machinery is essentially frozen. It does not tolerate horizontal gene transfer. Other less essential genes do, but now you have something that is very individual, which is a part of every domain of life and a part of every species, which is the type of translational machinery it has. And so then you have the descent of individual organismal lineages. In this phase of life, you have a network. That's what reticulates means. It means a network. So you don't have any notion of phylogeny. What happens here are communities that vary in descent and not individual lineages. So you have a community of organisms that are presumably very fragile, maybe even a lot of endosymbiosis, and genes and maybe other body parts are being transferred between them, leading to a very rapid evolution of the genetic code, which parallels the rapid evolution of antibiotic resistance today because of our horizontal gene transfer. Okay, so this makes us some suggestions about the prevalence of horizontal gene transfer back earlier in time. We can also test some of these mechanisms by doing experiments to look at detailed phylogeny of different types of cellular machinery to see if we can understand life earlier than the last universal common ancestor. That's what we're doing at Illinois. And here are my conclusions. Biology cannot be understood from snapshots. You have to think about evolution. In less than 1 billion years, life began and evolved the modern cell. Very little significant evolution of the cell after that. The cell's genetic code is universal and optimal. How did it get like that? It had to be from a collective phase of life. And we can probably find more data to be evidence of this collective state of life, and that's what we're trying to do at the moment. So that's what I wanted to tell you about. I'll be happy to take more questions.
So I think if you look in genetic code repositories, they have about 40 codes that they describe, and there's this amber codon, where a substantial fraction of bacteria uses that. So that doesn't quite go with this universality, does it? I mean, it's the same code, but there are variations of it.
So the variations are very small, and all the ones I know about. So I know about 24 different variations, and they're mostly mitochondrial.
I think in bacteria you have, for instance, this amber codon, which is turned to the stop codon. That means if you use standard gene calling on these kind of genomes, you will get very short genes, much shorter than they actually are.
Right. But it's still basically the same. I mean, it's not a very different code.
Yeah, but it's slightly different. It would be interesting to see how that does in terms of your metrics, how far these codes are.
I don't. In terms of the metric, we know that the canonical code is not the optimal code. So it's certainly easy to. What?
Which is the optimal?
it's not the canonical one. It's close to optimal, but you can find, if you look hard, you can find codes that are better. So, but for biology, it doesn't need to be the optimal code. It just needs to be, very good and good enough. So I doubt that we would see very much difference between what you're saying, but but that would be checkable. So that's a good point. Thank you. Yeah.
But you can actually use the simulation to produce an optimal code.
You can, yes.
And how far is it? It is the closest.
That's why if you go back to your diagram, right, so you had this sort of asymptotic, but like a 2 or something, right, presumably, right, or something like this.
No, that wasn't the number of codes, that was the difference.
Yeah, the difference,
but it eventually
yes,
goes
yes.
to 1. Yeah.
Right, but I mean, so, okay. But you could push it a little bit further down.
You can get more optimal codes. There's no question about that. I don't think there's a law that says it has to be the optimal one. Most things in biology are not optimal. They're just good enough and to.
So it was a frozen lesson in a way, because at some stage it was good enough that it could operate, and so then it was frozen, right?
Well, I would say it a different way. I would say that it has got to a fixed point of the coevolutionary dynamics, and it's very close to that fixed point right now. But it's not just some crazy random thing that was just sort of thrown together, which is what the straw man hypothesis is.
But the model doesn't explain why this mixing thing stopped. Right?
This model doesn't. No, what I've shown you doesn't. I can make arguments for why it would stop, but all the and there's several of them. You can use game theory. You can sort of wave your hands and say, well, once the machinery becomes very locked in, it becomes very hard for any new components to make it any better. So you can do things like that. But all those arguments, in my view, put in the answer. So we're still trying to think of a way to test how one can understand this transition. So there's no shortage of argument, but I can't give you a very compelling mathematical reason why it stopped. And in fact, we know it didn't stop for some things. So the question is, what does cut it off? What I think is basically it becomes so complex that once it becomes very complex, it just becomes harder and harder to change because it's so complicated. So I think that's most likely the reason. I mean, we think of the code, or the fact that you can even talk about translation from a code's perspective is very surprising. This was George Gamow's great idea, that you can take all this biochemistry and just say, who cares about biochemistry? In the end, all it does is this abstraction. It's very surprising that it does that, but there's so many different proteins and things involved that it just becomes very complex and very difficult to imagine that at some point you would get a gene that would improve it. So maybe it's simply that. So we can speculate about that, but I haven't answered that. You're right. It's a bit like the inflation. When inflationary cosmology was developed, what was hard was to figure out how to make the exit from inflation. So it's a similar type of thing.
So how comes this set with scenarios where you assume that at the beginning we probably had fewer amino acids and
Right.
started out with like 5 or 6?
Right. So the question was, what are scenarios where you say initially there were 4 or 5 amino acids and then gradually more and more amino acids were added to the code? So I think that it went slightly differently from that. I think the code grew not by adding things, but by going inwards. So what I think was that if you look at the codon table, I suspect it was always triplet codons, and I suspect that what happened was that initially different regions of the code table coded for sort of equivalence classes of amino acids. So as long as you had something like one of these things, that was good enough. And as life became more and more complex, those equivalence classes were broken down into more precise specifications. And so that's how the amino acids weren't sort of added to the code, they just became necessary to be translated for that particular point in the codon table. So that's my answer. So I don't think you had a machinery and then it sort of So okay, now we're going to do version 2.0, we're going to add in another 5 amino acids. So I think that they were sort of all there at the beginning, and then gradually became more and more specific. That's the picture that this says.
I didn't really understand this polar index or what
Yeah.
it was.
Yeah.
What's the basis for it? Why is this relevant?
Why? I don't know.
What exactly is it?
You mean, how was it measured?
Yeah.
The way it was measured was this. Carl Woese took amino acids and water pyridine mixtures. He did paper chromatography and measured how far they diffused. He then plotted the distance that the amino acids diffused as a function of the water concentration. And when he did that and plotted it on log paper, he found that he got straight lines. So from those straight lines, he could produce one number, which was the slope. And the number is what he called the polar requirement.
But basically hydrophilicity, right? Yeah.
So we tried to understand what it is. So what we did with Zan Schulzen was do molecular dynamics simulations of amino acids and measure the correlation function for the end-to-end distance, and we found that there's a number you can extract from that, which is basically the decay of the correlations. That number tracks the polar requirement measured numbers very well, except for tyrosine for some reason. And so we can make a version of the polar requirement that you can measure in a computer. But the rationale for it is, well, Carl woke up one morning and said, I think early life was in these, this was the sort of chemical environment of early life, and so I'm going to sit down and measure this thing. So nobody really knows why it works so well. But it obviously is telling us something about early life, but that's a job for somebody who has more background in biochemistry and chemistry than I do. So,
Wasn't there something with a pyrimidine in the definition, or something with the nucleoside?
Mhm.
Or when you're on your slide?
yes. So I mean, there's well, in terms of the here we go. So this is how chemistry people interpret it. That's what it is. That's what I mean. I think of it as basically the difference between this and this. But why it does that and how nobody really knows. So
Is there any evidence that if you change in a given protein an amino acid with something that has a similar index, that the protein is less affected than if you would exchange it?
Yeah. So people have done experiments. For example, Paul Schimmel has done experiments where they tried to take translation back to make statistical proteins. So that people, so we know how to do that. So that's been done.
And so that really suggests that this polar requirement is a good metric of error.
Yes.
Or whatever.
I'm not really very well qualified to interpret the chemistry of what these people do, but they will say yes. They use this to try to make experimentally situations where you have statistical proteins.
So which gene will be the word application?
Each what?
Which gene will be the word application? Because you need the winning application that fits in the system, and this is make the operate as one.
Well, I would say the canonical genetic code is like the Windows or the Word.
What will be the word? Yeah.
What will be the word?
Yeah.
I don't know.
Which gene?
I don't know. 16 has 5%, well, 18, I don't know.
So thanks.
Okay.
So we have some beer in the fridge. And
There's what?
Crisps.
Beer.
There's beer and crisps,
Okay.
which we can have back in the couple of days.